Okay, hello everyone, I'm Viveka and I'm from Group 13, and our article's topic is a multiple overseas dispersal in amphibians. And what we mean by amphibian is a cold-blooded vertebrate animals which have gill and also lungs, which uh, allow them to live on a land and in a water. And this group of animal is a key group in historical biogeography because they are said to be unable dispersed through saltwater barriers, and they are extremely sensitive to osmotic stress and do not survive in salt water. So amphibian is an excellent model for vicariance scenario as explanation for general biogeographic patterns and major biogeographical hypotheses that have been influenced by occurrence of endemic amphibian on islands. And what I meant by vicariance is a geographical separations of a population, which is typically a physical barrier such as a mountain, a river, resulting in a pair of a closely related species. And the distributions of the amphibians is widespread in many archipelagos such as Philippines, Sunda, Caribbean, and Pacific regions. And what I meant by archipelagos is a group of islands which is separated for the land masses, which is considered as isolated from other land. So these islands is made up least partly by continental fragments, and the distribution pattern of this amphibian in this article is explained by the Vicariance biogeography as I mentioned before, and this article shows evidence of overseas dispersal by this frog in Indian Ocean region based on discovery of two endemic species in a Mayotte. So basically the objective of this journal is to represent a report on recent discovery that the Comoro frogs, previously undescribed species endemic to Mayotte. Mayotte is an archipelago in the Indian Ocean between Madagascar and the coast of Mozambique.
There are four methods that have been used for this research, which is the taxon and gene sampling, the DNA extraction, amplification and sequencing, the phylogenetic analysis, and the last one is the molecular clock calibration. For the first method, the purpose of this method is to understand the distinctness of the Comoro frogs, to understand the phylogenetic relationship among mantellids, and to understand their relationship to other frogs group from the Indian Ocean region. For the second method, the DNA was extracted from the eternal preservation and sequenced on ABI 3100 and ABI 377 automated sequences after direct amplification using the primer from the previous study or the primer from this work that was developed. For the third method, which is phylogenetic analysis, the genes were submitted to separate and combine analysis with PAUP after exclusion of all gap and hypervariable region of the rRNA and tRNA genes and the third positions of the cytochrome B, which are known to be fully saturated at the level of environ families. And the last one, the oldest available age of the Mayotte provides a calibration of the age of the split between the Comoruan and the Malagasy sister species.
Results 8 talk about the discovery and relationship of Comoro frogs. Surveys were done on 3 Comoro islands, which were Mayotte, Moheli, and Grande Comoro. However, only Mayotte has amphibians. In Mayotte, they found 2 distinct frog species that corresponded with the previous recorded Mantidactylus granulatus. However, both frogs are morphologically different from M. granulatus. Other than that, both species live in similar habitats and are common in Mayotte. Using molecular phylogeny, it was found that Comoro frogs were deeply nested in the Mentelidae, where the cladogram placed them as sister species of the Malagasy emuitae and of the B. dolioti.
We'll talk about the first discussion for our article, which is the first evidence for frog overseas dispersal to oceanic island. They estimate that under the sea levels, the Malagasy northwestern coast and Mayotte were still separated by a linear minimum distance of more than 250 kilometers and by ocean depths of more than 3,400 meters below sea level. Many new species of frogs have been described from Madagascar in recent years, but most are morphologically close to known species and many were already present in historical collection. The Philippines harbor a largely endemic frogs fauna but were probably entirely submerged during the Oligocene, leaving subsequent dispersal as most probably origin of these amphibians. The Philippines were probably connected to the Asian mainland through land bridge in the Pleistocene. Consequently, the discovery of endemic Comoro frogs is the first evidence endemic amphibian species occurring fully volcanic and oceanic islands, and thereby the most reliable evidence for overseas dispersal in this vertebrate class.
So for this part, I'm going to talk about the B section of discussion. The title of B part is Gondwanan versus post-Gondwanan origin of rhinoid frogs. So according to molecular clock datings, which is done by the scientists, they have found that at least 2 cases of transmarine frog dispersal happens in the western Indian Ocean. The first case is happens in hyperolite frog. So the movement of hyperolite frogs, which is the heterocellus, moved from Africa to Madagascar. And then there's another frog, which is the Tachynemis. It moves from the Madagascar to Seychelles. And then there's another event where there are 3 dispersal events happens from Asia to Africa in a tertiary period. So the dispersal of this is estimated during Oligocene and Miocene epoch. Another part of the research is that they found that within the Mentelidae frogs, namely the Comoro frogs, the Comoro frogs are deeply nested in the lineage of arboreal, semi-arboreal, and pond breeders. That means they live either on trees or semi-arboreal. And the same case happens for hyperolite frogs, which is the Malagasy and Seychelles frogs. One of the important adaptations to cross the ocean barrier is to have tolerance to xeric environment for the frogs. This happens when the frogs are able to migrate via the leaves of the rafting trees from Africa to Madagascar or from Madagascar to nearby islands. And to be able to do so, the frogs need to be able to withstand a very long period without water. So this is called the tolerance to xeric environment. So they found out that the ocean currents at the present state favors a rafting from northwestern Madagascar to Mayotte, the Mayotte is between Madagascar and Africa. So the current ocean flow favors from Madagascar to Mayotte, while during early tertiary periods, the ocean current favors the rafting from Africa to Madagascar. So this can explain why the frogs are able to travel via the leaves or the trunks of trees to Madagascar with the flow from Africa to Madagascar. The conclusion is the Malagasy fauna is contributed by the dispersal from Africa, most probably through the currently submerged islands, which act as the stepping stones for the dispersal of animals from Africa to Madagascar. And then another factor might be due to many dispersal events, which is from Madagascar to Comoros Islands and to Seychelles, and also from these islands partly back to Africa. And all of these factors contributed to the diversity of terrestrial and freshwater animals in the Indian Ocean region.